US is amazing with its visa situation. I could now go, but there's always an issue for me to come back.
Yeah.
And it's always painful.
But in fact, I was supposed to go to India early March. I was supposed to go to different cities in Pune, Mumbai, New Delhi, and then Bangalore, and it was all,
like a whole tour.
Yeah, we postponed because of the restrictions, and they were just lifted, like say, end of March.
Yeah,
and that is the reason why I eventually managed to go to early April.
Yeah, I will try to go because I feel like my family has probably already forgotten my face now, so should go.
Well, especially the hair color.
Yeah. Okay, we have like 2 minutes more to go.
Sure.
And then we'll start. Okay. Jen, it's all good. I know Jen must be killing herself for not able to talk today. So are you okay with us keeping it on YouTube, Thomas? Because we usually have like 400 people watch it later.
Sure, no problem.
Sounds good. Okay, so it's almost 10:59. And while I introduce, I'm sure people will trickle in more. Okay, so welcome everyone for another session of Cell Migration Seminar. Today, it's an honor and privilege to welcome Professor Thomas Lecuit for our seminar series. I'm personally super thrilled to do this because if people don't know, I did my PhD with Thomas like 4 or 5 years back now. So Thomas did his master thesis with Professor Claude Desplat in Rockefeller University and went to EMBL Heidelberg, Germany to do his PhD with Dr. Stephen Cohen. After his PhD, he came back to US to do his postdoc with Professor Eric Richoz at Princeton. And in 2001, started his own lab in EBDM Marseille in France, where his lab focuses on understanding how information and mechanics sort of work together to generate shape. And they do that using Drosophila as a model system. He became professor at Collège de France in 2016, and he is also director of Turing Center for Living Systems in Marseille that focuses on complexity and self-organization in biology through contribution of physics, computer science, and mathematics, which I think is a very interesting thing. Today, he is going to talk about collector cell motility associated with wave of embryonic tissue deformation. Thank you, Thomas, so much for accepting our invitation, and the floor is all yours. And we will read you the questions, as like some people interrupt maybe in the middle a little bit, but mostly it's in the end of your talk. But if you want to stop around 20-25 minutes, we can do that as well.
Yes, so thank you very much, Ankita and Jennifer, for this great invitation. I'm indeed very excited about being with you. This is one of the new ways in which we can actually reach out to people far away. So welcome everybody, morning, evening, maybe for some people. So I would like to indeed, in the theme of motility of this series, my lab is typically not working on cell migration, but 2 different projects in my group have been sort of converging on topics of cell motility. And so I chose one of them today, which is what happens in the embryo. So the title will become clearer as I go and tell you through our thinking about morphogenesis more generally. So first, let me just emphasize that people who have done the work, so I will be talking about the work, some of it published, others unpublished, of 2 great people in my group. So Claudio here, when we were in retreat in Rome a few years ago, and Anais, who is a physicist, and Claudio is a biologist. And so they teamed up and they've been working on this process of embryo morphogenesis, which I'll talk about later. And other topics in my group revolve about questions of shape in neurons, which would be another topic of motility in a sense, as well as how we encode size in embryos. So let me go straight to how I would like to sort of introduce this broad problem of morphogenesis, which my lab is interested in. So as we all know, cells emerged something like 3.5 billion years ago. And in stromatolites, which you could see on the right as they look today, and on the left, you have a fossil. if we were to look at what happens then 3.5 billion years ago and since then, we'll be looking at something which looks like that. Bacteria dividing at a pace of 1 cell division every hour or so, depending on condition. And we take for granted that this process of cell division is essentially trivial, that what happens is basically we understand it very well. But in thinking about this process, I would like to ask sort of the broader question of how cellular organization transmitted from one cell to its descendants. That is, of course, taking place every time a cell divides, but it's also taking place every time a new embryo is formed from a fertilized egg. And so if we, let me go through what could be sort of a thought experiment. So an E. coli cell is a simple cell compared to eukaryotic cells, but already there you have quite an exquisite organization in the form of macromolecular complexes. Think of the rotor and the flagellum. And the chemical composition is also quite complex. Of course, you have a few million proteins, a few tens of thousands of ribosomes. And the question is, if we were to actually grind a cell to complete chemical homogeneity, and I mean by that in the hard way, we would actually even break down the polymers into single amino acids or nucleotides. And if we kept ATP at good concentrations, if we waited and observed, what would we see? Of course, if the polymers were broken down, although the chemical complexity would be even far greater than it was over 3.5 billion years ago, we would not be able to resume life, although all the chemistry is there. So we need some level of organization which minimally is present in the form of polymers. So now let's think about the soft way of doing the experiment. We grind down to chemical homogeneity, but we keep polymers, like we keep DNA, we keep mRNAs, and we keep proteins. We have ATP, we have everything. In principle, would we see a resumption of life in the sense of dynamics, organization, and formation of a new cell? Well, typically the answer is no. Even though we have all the chemical complexity and even some part of the history in the form of polymers that witness sort of the history of E. coli to this stage, we will not be able to actually reform a cell and the complex organization of an E. coli cell. So in a way, life is not, and the transmission of organization is not 100% contained in DNA. Every time a cell divides, there is a heredity of structures, a structural heredity, which is a form of some complexes or structures like a lipid membrane bilayer and things like that. And if this is true in E. coli, it's even more so the case of any eukaryotic cell. So the point and the reason why I actually introduced this question is because it leads me to the problem of heredity in the context of cell division, cell organization, and development. And so in thinking more generally about the questions of information, which really underlies the process of emergence of patterns and shapes, we're very familiar with a kind of heredity, which is a genetic heredity. And of course, that actually encodes a complex biochemistry, which gives rise to itself to mechanics and means to actually control shape at the cellular level as well as at tissue level. So these are two important modules of information. But sort of the missing part here would be a module of information, which we could call, I'm stuck here. Excuse me, my computer is blocked, so I need to
no worries.
Basically, yes. So the third module of information, which is connected to my...
Your thing is not shared yet, Thomas.
I'm sorry.
You want to reshare it, I think?
Yeah.
Perfect.
So the third module of information is geometry. I mean by this, in the very broad conceptual sense, any structure that is one inherited, and is typically what I referred to when I was going through the thought experiment of an E. coli cell that actually transmits some structures for life and organization to be further established every time a cell divides or every time an embryo is formed de novo. And so that is part of an heredity. Heredity is a form of DNA and chemistry, as well as is in the form of many structures and geometry. So we have in mind now these three modules of information: biochemistry, geometry, and mechanics. And we can ask ourselves is, how are the three modules of information actually organized and interacting with one another to underlie the emergence of any structure. I don't have the time, but I'm sure you're familiar with in what sense do I mean that biochemistry, mechanics, and geometry are three modules of information. Think of biochemistry, information is basically the statement about defining length scales and time scales that actually would be required to organize pattern within a cell as well as within a tissue or an embryo. So biochemistry can do it by diffusion and reaction diffusion processes, for instance. But mechanics can also do it. The properties of a system would actually endow it with the definition of length scales depending on viscoelastic properties, for instance, of its constituents, whether it be a cell or a tissue. And geometry is a statement of initial as well as boundary conditions that allow for different parts of a cell or tissues to be connected non-locally by means of mechanics and chemistry. So yes, really, the three modules of information really set up particular processes. And so my talk today will be basically dealing with how the three modules of information actually underlie complex morphogenetic processes in the embryo and how actually cell motility kicks into this context. So at this point, I'd like to introduce two modes of information flow, which we are in a way familiar with. And these two modes of information are in a way ideal. They are sort of reference in terms of our thinking. And the first one is what we sometimes refer to as a program. I mean by that, a statement of hierarchy in the flow of information that basically uses as initial conditions, what is inherited, whether it be biochemistry or geometry, and that would deterministically predict or control actually mechanics, say contractility in cells and tissues, and from that you could actually predict specific morphogenetic outcome. And I'll go through illustration of that specific modes of information flow in a few slides. But another one, which is very different, is referred to as self-organization, basically abandons completely the idea of hierarchy. And through many feedbacks and stochastic processes, actually allows for the emergence of patterns from initial homogeneous isotropic conditions. And the interactions may be simply local and actually could give rise to the emergence of complex patterns, such as on the right termite mound. But thinks of shearing instability as another way of such self-organization processes. And so we're interested in the way that self-organization and program morphogenesis actually underlie any morphogenetic process. In what sense is any process governed by deterministic hierarchical organization of information as well as self-organized processes? In the context of fly embryos, which we study, a well-known example of process that actually underlies the idea of a morphogenetic program is what we refer to as pattern formation. The concept of positional information is basically sort of the chemical concentration of a species, of a bunch of molecules actually defines the Cartesian coordinates of a system along the anterior-posterior dorsal-ventral axis, and that's how you actually define the coordinates of cells and their fate. And so what people have characterized over many years is the fact that such positional information provides initial conditions that actually sets and controls cortical mechanics. I mean by that typically contractility, which then is going to govern cell behavior, cell dynamics, and from then actually tissue morphogenetic processes. In the context of, for instance, extension of a tissue, so on the bottom left, you can see two types of very general classes of tissue morphogenetic processes, namely tissue extension on the bottom right and tissue invagination on the bottom left. And so these are basically taking place in specific regions of the embryo because of the positional information. And they're both driven by a control over cell mechanics, cell contractility. So on the right, for instance, you can have a tissue in which cells actually are going to change the position by the process of so-called cell intercalation driven by plano-polarized cell contractility, which I'm not going to talk about. But more important for the context of my talk would be tissue invagination. This process, which is occurring in very many organisms, actually requires cell apical constriction, which is a reduction in the apical surface area of the cells, which is driven by the apical accumulation of actomyosin contractility, which you can see on the right in the little movie from Alan Martin's lab at MIT, where they report myosin 2 GFP dynamics as a function of cell area reduction. And so here, the process of apical contraction actually determines a collective invagination of the tissue. This is now genetically programmed in the following sense. If you look through the embryo, and you can see here like kind of a slice through the embryo, you can see that the ventral cells marked in red specifically express two transcription factors called twist and snail, which are sort of switch factor in the sense of a genetic switch that actually activate a pathway that leads to a reorganization of cell contractility. The cells are basically born with a basal contractility marked in orange here. And in the region where cells express twist and snail, apical contractility will take place, giving rise to apical constriction, basal expansion, and that collectively give rise to tissue invagination, as you can see on the bottom right of this graph. Though they really work as switch factor, because if you look at the pathway that leads to myosin to activation here, in green you have all components from GPCRs, Rho, ROCK, and all these molecules which are expressed everywhere in the embryo. But selectively in those cells marked in red, there's a ligand to the GPCR pathway as well as other cofactors that actually boost signaling, switch it on, and allows for the activation of myosin to contractility, specifically in the apical surface of those epithelial cells that are in the ventral area. So this is a good example of how positional chemical information switches on cortical mechanics and induces cell deformation and collectively tissue invagination. This is the example of a program. Now, I'd like to sort of take this on board and to sort of revisit this context, this process in the context of another invagination that is taking place in the fly embryo. And so here you have a movie taken by Anais and Claudio a few years back, where they were looking now at 2-photon microscopy through the embryo. And what you can see is actually a sort of flow of cells that actually rotates toward the dorsal part, which is here at the top, an invagination that builds up, and then sort of further propagation of this deformation to the right. And so I'll be addressing specifically the process that is taking place now, which is sort of a propagation of this deformation to the dorsal part of the embryo. I will not be talking about the flow, which is, however, a very interesting process. So if we look at this tissue from the dorsal part with the spinning disc confocal microscope, you will see now the cell outline marked in purple. This is Ecuador and GFP. And if you look at myosin, which is marked in green, you first see an activation of myosin 2 in the apical surface of these epithelial cells in a sort of a disc, which is the posterior of the embryo. And this is actually in a very specifically defined spatial domain called the primordium, which is a bit similar to what happened in the mesoderm that I showed you before, where actually there was an invagination specifically in the ventral midline of the embryo. Now this is in a different geometry, which is sort of the posterior pole of the embryo. But soon after that activation in the primordium, you'll see a process which really struck us as very, very different, which is sort of a wave of activation of contractility sweeping from left to right, meaning from posterior to anterior through the embryo. And so the movie will loop, and you will see a number of things which I document step by step. First, cells that are marked in yellow are at some distance from the primordium, and they expand the apical surface, then activate myosin 2 in green, and subsequently are even sort of engulfed into the invagination, which actually propagates to the right. So we have sort of the linear propagation of 1, cell deformation, 2, myosin 2 activation, 3rd, the collective tissue propagation, invagination. And of course, this linear propagation is very surprising because the embryo, the fly embryo, has been known for many years as a system in which actually pattern formation arrives quickly at a steady state, sets up some domains where particular processes are going on, such as the one that I told you about before, namely mesoderm invagination. So here we're sort of witnessing a very dynamic process, which is at odds with the classical view of a positional information dependent control over cell mechanics. So we have these two domains, a primordium and a propagation zone, as we call it, and we'd like to understand the process that underlies wave propagation in the system. And so these are still images, which I'm not going to comment upon more, but now we could sort of study this quantitatively by looking at the movement of the wave in the co-moving referential of cells that actually are moving with the tissue, because in a way, there are two compound processes going on. One is a flow, namely the primordium that you see on the left at the top is actually moving as a result of sort of tissue flow to the right. But in this moving referential, you could look at the wave propagation of myosin to contractility, and that is what is plotted in the chymograph on the right. We use as a referential sort of the moving referential of the primordium that flows to the right. And you see that the propagation of cell deformation at the top and cell contractility at the bottom is actually linear. It has a characteristic linear propagation. Here's actually a zoom of this particular process where you can see myosin 2 contractility in green and the cells outline marked by ecoderion in purple. I'll come back to sort of gradually at sort of the description of this process and we'll progressively move to actually even cellular description of that wave propagation. But you could appreciate that there is sort of a gradient at any moment of propagation of contractility from the edge of the invagination where it's highest levels to more anterior region where it's actually the lower levels. And so we'll keep track of the gradients because it is a wave. What we see at any moment in space in a gradient is actually a reflection of time of activation. So here's sort of a cartoon of the embryo in which this is taking place. So I talked to you before at the beginning about the mesoderm in red, which is sort of taking place in the ventral part of the embryo. This is a very fixed domain, well-defined, where invagination is taking place. Now in the posterior, the primordium in blue is triggered by other kinds of transcription factors that actually define this domain and induce, in a very similar way to what happened in the mesoderm, myosin 2 activation. By activation through the production of a ligand to a GPCR, G proteins, in particular G alpha GTP, which is going to activate through ROGEF2, the Rho GTP, which activates ROC and then myosin 2 phosphorylation. And that pathway is the same in the ventral part, in the mesoderm, and in the posterior part. The only difference being the switch factors which actually induce FOG transcription. And you could actually replace potentially one with the other. FOG is necessary and sufficient to fire activation of myosin 2 in any of these epithelial cells. So having that in mind, we first considered a model, which I'm going to basically quickly falsify by not showing you any data. But the first thought we had is that if the system is primed genetically by the transcription of switch factors which induce FOG in the primordium, might it be that FOG is actually transcribed in a wave through the tissue? We could clearly show that this is not the case because after the system is sort of induced, after the first domino, if you wish, of the series falls, then we could block transcription altogether by injection of, say, alpha-mannitine, and the linear propagation of the wave was taking place normally. So clearly, although the system is primed genetically by transcription of the constituents, in particular FOG, once it's on, it sort of progresses linearly without any further input of transcription. The second model was actually one based on the knowledge that FOG is a secreted protein, a ligand, which actually diffuses and can activate potentially the GPCR at a distance. So might it be that sort of diffusion or transport of FOG through the tissue in the vitelline space might underlie the linear propagation of the whole tissue, of the whole activation of myosin 2. Well, in fact, this is not taking place. And I'm not going to go to any data. This has been published. I'd like to go just through a simple toy model that is sort of was a means for us to already tell us that it cannot take place because there are particular signatures of such diffusion system. And so the kind of toy model that I'm referring to is one in which you have simply stated the production of FOG, its diffusion, its local degradation, and its capacity to activate myosin 2 and induce stress. And that would basically propagate in the tissue by diffusion of FOG. And the salient feature of that is that the rate of activation would decay as a function of time and space. And there would be dependency on the production rate of FOG production, which basically were all falsified by experiments. So the bottom line is that, playing with dose of FOG, patterns of FOG, we could show that such a diffusion diffusion system is not underlying the linear propagation of the wave through the tissue. It's required for its induction, but it's not for its sustained propagation. So that led us to the proposal that the process might be actually driven by some kind of mechanical process, which we have, or mechanochemical process, which we have to characterize. So very generically, the idea would be that local contractility, say in the primordium, would have a means to communicate with more anterior cells. And the means by which it communicates, this sort of represented by this red arrow, is going to take us some further development, because it's not as simple as I'm contracting in the primordium, and that actually simply mechanically propagates to the neighboring cell by some simple active stress that strains the neighboring cell. It's actually much more interesting than this. So let me go through this. So first, using again the sort of simple toy model that I used for presenting the principle of diffusion, let me now just modify this model by saying, okay, let me now not allow fog to diffuse at all. It's going to be produced and degraded, similarly to previous simulations. Now I'm going to do two modifications. It's not going to diffuse. However, I'm going to allow the stress to feedback onto myosin II concentration. And so that simple feedback actually gives rise to a trigger wave dynamic, an undamped sustained linear propagation of activity, as you could see in the simulation and in the chirograph on the right, which was a way for us to simply check that such a model would actually broadly fit with the simple phenomenology of a linear propagation and damped activation of myosin II to the whole tissue. So the core tenets of the model is that there is a feedback of stress onto myosin II, or that a consequence of stress, such as strain of the network, actin network, and myosin II. So in fact, this is not coming from nowhere, because it's well known that myosin motors, in particular myosin II, is stabilized by strain. So that's a very plausible scenario. So let's look at this. So we did an experiment, which is shown here, in which we could actually look at the co-dynamics of Rho GTP sensor, which we're looking at the top here. And you could see, say on the left, top left, top right, we see the localization of the Rho GTP sensor. I'm going to activate the movie in a second. And at the bottom, you have myosin II labeled with cherry. And so if you focus on the left, you will see in the movie that, they both move together as a wave perfectly synchronously. But then on the right, we'll do an experiment where we inject at a given time point a ROCK inhibitor, which is going to give rise to the dissociation of myosin II from the cortex within a few seconds. So effectively, myosin II will no longer be at the cortex. And we will ask, how is the propagation of the Rho GTP sensor taking place in this context? And so on the left, you see the control. You could see that you have the co-dynamics of both Rho GTP sensor and myosin II. And on the right, you see that as soon as myosin II is inactivated through ROCK inhibition, we see that myosin II is gone. And interestingly, now let's look at the Rho GTP sensor. You'll see that the levels abruptly go down to low levels and remains at these low levels. So there are two phenomena taking place together. One is that the propagation of high-level Rho GTP fails, and the actual linear propagation of the whole deformation of the tissue is also stalled. What you are ending up with is just low level of Rho GTP, which probably reflects, we think, and I will not talk about that, what fog diffusion is possibly doing by sort of activating GPCRs and activating low levels of Rho GTP, but not more. And so in a way, the linear propagation of the wave requires a feedback of myosin II contractility onto Rho GTP itself. So that sort of feedback is exactly what we suspected might be going on to explain the linear propagation of the system. So more than that, if you look at a given cell, once you activate the ROCK inhibitor, so within seconds at the bottom, myosin II goes away. And you could see that even Rho GTP, which normally forms this aggregate, this aster, is actually dissolved into single units that actually disperse in the cell. So we fail to sustain myosin II and Rho GTP, Rho GTP once myosin II is actually deactivated, myosin II is inhibited. So that gives us, leads us to conclude two things. One is that there is a positive feedback that is taking place at the single cell scale that allows to sustain myosin II, Rho GTP and myosin II together. And second, the fact that there is a spatial coupling that allows this activation to be transmitted from one cell to the next progressively by some sort of spatial coupling, which we argue from this experiment have some mechanical component. And so that is typically what now we need to nail down more precisely. And as I said, what it's not going to be is a simple transmission of strain in the neighboring cell, which is going to activate myosin 2. We'll have to take into account precisely what I refer to as geometry. So sort of shape of the tissue, its curvature, its shape is essential to actually account for the propagation. Mechanics and chemistry are not enough to explain the propagation. And so that's why from now I'm going to look at the tissue from the side, exactly as this drawing is showing, with the dorsal part of the tissue where the wave is taking place and the invagination. So this is now the time when we actually decided to look specifically at two-photon imaging to characterize the tissue geometry. And so you can see at the top a movie of this tissue where we can see the columnar organization of the epithelial layer. And we can see that in the white box region, there is a sort of a hinge that forms. The cells are actually bending toward the posterior. In black is actually the nucleus. And then the curvature goes on and this propagates to the right. On the right, you see sort of time series, a few minutes apart of this very process. And if you focus on the cell that is marked in yellow, you could see something interesting, that the base of the cells translates to the right, but the top part of the cell is actually almost immobile. It's actually as if it was stuck to something. And what I'll show you later on is that it is indeed stuck to sort of an invisible layer called the vitelline membrane, which is like a stiff ECM matrix that actually sort of encapsulates the whole embryo and protects it against, and to create sort of a local chemical environment. And so at the bottom now, I'm going to draw sort of a cartoon model, which is going to be sort of the basis for a proposal of how the propagation goes on. And so I'd like to begin with the, so my arrowhead at the bottom shows it. In this invagination, all the cells are contractile. You can actually see it with myosin 2 labeled at the top. All the cells have undergone apical constriction, and as a result, they give rise to an invagination. And whenever you have an invagination, this gives rise to a compression of the whole cytoplasm to the right. This compression is going to do a number of things on top of actually flowing the whole cytoplasm to the right. Is also giving rise to some movement to the top as well. And so this arrow shows in green how the whole cellular content, multicellular content, is actually shifted toward the dorsal part against the vitelline membrane. And so I'm going to say a number of things which I will then test experimentally later on. But yes, this tissue is basically lifted against the vitelline membrane, where actually the cells are going to adhere to the vitelline membrane. And this is going to give rise to myosin 2 activation in a way which I will address in the next part of my talk. And eventually there's going to be a de-adhesion or a detachment of the cells from the vitelline membrane by the very contractility of the cells in orange, which actually are pulling perpendicular to the vitelline membrane and detach the cells. So this whole cycle of contractility, compression, adhesion, myosin 2 activation, de-adhesion is giving rise to the self-sustained mechanochemical cycle, where actually the whole geometry curvature of the tissue is an essential ingredient of that process. And I'm going now to document that in more detail and test it in the next few slides. So first, the reason why it gives rise to a self-sustained process is that once a cell is activating myosin to the top, it's going to be detached from the vitelline membrane by the contractility of its neighboring cells in the furrow. They will actually feed the furrow. It will sort of add new cells. And because you add a new cell to a furrow, it's going to have to move to a more anterior region to actually accommodate this new cell. And that gives rise to a propagation of compression to an even more anterior region, more adhesion, more contractility. And so the system would actually roll in on its own. And so to test this, just look at what happens when you look on the side at the whole process and inject a ROCK inhibitor. You see that there is an instantaneous block within seconds of the whole process of not just myosin 2 activation, but also curvature, detachment of the cell. So every single process that is documented in a color code, contractility, compression, adhesion, myosin 2 activation, and de-adhesion, are all parts of a wave. They are all following the whole wave dynamics together because they are mechanically or chemically coupled to one another. And so now let me show you some evidence that indeed the cells are being lifted against the vitelline membrane due to the compression associated with contractility in the furrow, and thereby engaging with adhesion to the substratum, which turns out to be outside the embryo in the form of this vitelline membrane. So on the left, what we've done is that we've injected dextran fluorescently labeled in purple. And you can see that it's sort of filling the invagination on the left with a bright color. But now precisely in the region where myosin 2 is activated, you can see that now dextran is excluded from the sort of vitelline space because it's basically being pushed against the vitelline membrane. So that really told us that, yes, these cells where myosin 2 is induced are precisely brought in contact with the vitelline membrane. More precisely on the right, you will see actually now a co-labeling of the plasma membrane in green and myosin 2 in white. And what you will see is zooming in on an embryo in which actually the cells, one of the cells is going to be sort of lagging behind. You see, I hope the movie actually goes through, but you will see that some of the cells are actually sort of being stuck to the vitelline membrane longer than they normally are. And they are then snapping back, sort of pulled by their neighbors collectively. So think of here this tissue as being a tissue that is going to be stuck to the vitelline membrane, it looks like. And normally all the cells sort of detach in a sequence, like you wouldn't peel a tape from another surface. But here a cell actually is going to be sticking more than its neighbors. And interestingly, I'll come back to that. Those very cells in which actually the cells are more sticking are also going to activate myosin 2 at higher levels. The white levels of myosin 2 is higher in those sort of hyper-sticking cells. So that convinced us that it's actually not just a sort of a friction, but a bona fide adhesion to the vitelline membrane. And it turns out that this is driven by integrins. Integrin, as you all know, is present in beta forms and alpha form. So the beta chain is ubiquitously expressed in the embryo. Every cell in the embryo expresses the beta chain, but the alpha chain scab, as it's called, is specifically expressed in the very domain where actually we know that myosin 2 is going to be induced in the wave. And so that led us to test the function of alpha chain. I'm not going to show you, but beta chain, although it's expressed everywhere, is only sort of targeted to the plasma membrane where the alpha chain is expressed because the dimer is required for its targeting to the plasma membrane. So in a way, I'll show you later on beta chain labeling with YFP, the endogenous locus with CRISPR, and you will see that it's being induced, recruited the plasma membrane precisely in this zone of adhesion. But now before we go there, let's look at the phenotypes of actually knocking down alpha integrin scab in the embryo. And I'm starting with a sort of a tissue level phenotype. You know, remember that we are trying to account for the fact that sort of these integrin might be actually responsible for the sticking of the dorsal part of the tissue where the wave of activation is taking place against the vitelline membrane. So the prediction is that, you know, given that there is a flow of the posterior part of the embryo here to the right, think of actually a moving tissue that is resisted by a adhesion to a substratum. You'd expect to have some kind of buckling, which you actually see in the wild type. And that when you remove this sort of stickiness or adhesion, then you will have a shallow furrow and a faster flow to the right, which is exactly what we observe in this alpha chain mutant. You can see that on the side view, there's a very rapid translocation to the right. And the furrow is actually very shallow to begin with, until eventually, so you see it's very shallow compared to the wild type, until eventually there's even backward flow going on. So at the sort of tissue level, this actually tells us that yes, alpha chain integrin is really required for resistance to flow of the posterior part of the embryo. So I don't know if my camera allows to see, but think of a flow to the right. If I'm resisting that with sort of an adhesion, I will have some kind of buckling, and that is what is responsible for the furrow in the first place. And now if adhesion is gone because I remove integrin, then it's going to slip through against the vitelline membrane. I will have a shallow furrow and it's going to move faster. So all of that is sort of exactly what we see at the tissue level. And let's look at the cellular level. We can see that now at the edge of the furrow, the cells in the mutant for integrin are actually showing much lower level of myosin 2. At the top, you have a control, and at the bottom, you have the scab mutant in which the levels are much lower, and this is being quantified on the left here. So the important thing is that knocking down alpha chain integrin is giving rise to a downregulation of myosin 2 and a failure to actually propagate a wave of high myosin 2 activation through the whole embryo. And so that is typically what we were expecting given our proposed model. And so at this point, let me sort of summarize at the tissue level how we understand the process of mechanochemical propagation of myosin 2 activation and tissue deformation through the tissue. So I sort of start at the bottom with this sort of model of a cycle involving adhesion, activation of myosin 2 against the vitelline membrane, de-adhesion, and then this propagation. In a sort of more conceptual way, you could look at the bottom. The cycle would go as the myosin 2 based contractility on the left would cause invagination, which is then going to cause compression, integrin based adhesion, and sort of this cycle of activation has long range effect because the compression actually is propagating through many cell layers, actually about 3, 4, 5 to the anterior region of the invagination. But on top of this long range activation in red, the invagination is responsible for the detachment of the cells at the edge, right at the edge of this furrow of this invagination. The invagination is going to pull the cells and detach them from the integrin-based region. And this has a much shorter range of activation. So now what I would like to do is basically address more specifically the mechanism of myosin 2 activation, which I've just told you is at this point integrin dependent. I would like to go through what are the elements that actually allow that activation to take place, and how do we understand this process of wave propagation at the more sort of subcellular scale. And so here's actually is a view of a single cell that is sort of at the edge of the invagination, while we know that the wave is propagating at the tissue level. And so if you look at the single cell, you will see that its apical surface is initially large, is broad, is sort of, stretched maximally along the anterior posterior axis. Myosin 2 is shown in green, actin in white. And you can see that the apical surface is sort of shrinking and shrinking. The reason why it does shrink is because the cells are sort of being, the apical surface is being unpeeled or removed from the vitelline membrane. And so as it does so, it goes out of focus and is sort of behind the focal plane. So that's why it becomes smaller and smaller. If you look at myosin 2, it does propagate through the cell in a wave. And it does so in a way that is very striking, which is sort of, is going through 2 or 3 pulses of, or actually asters of contraction, which are seen in the chymograph at the bottom. Here's actually in sort of an aggregate of myosin 2, which is then going to be followed by another 2 more anterior region, and then the third one to the anterior region. And so we ask, how is this subcellular wave of activation taking place? We argue that whatever happens within the cell is in miniature what happened at the tissue scale. And so we could sort of zoom in on this particular cell to address the mechanism of activation. And so if we look more carefully at this specific cell, we could actually look at what happens to the cortex of these cells. And so cells who people work on cell motility work with the sort of ventral part of the cell, the basal part of the cells, again, a substrate. Here in the embryo, the only substrate is actually the outer, the vitelline membrane. So that's sort of a referential, a mechanical substrate upon which cells can actually, that the cells could actually recognize, adhere to, and do things. And here what they do is that they use the apical surface as a zone of contact against the vitelline membrane. And so the cells expand the area. They do so because they say, in a way, the cells are sticking to the vitelline membrane. And so any bit of the cortex that connects with the vitelline membrane is going to be immobilized. And so to convince you that this cortex is immobilized, that as the cell expands its surface, you could look at sort of a fraction of the actin network shown in white. And you can see that the trajectories of these speckles are actually become more and more sort of immobilized. They sort of jiggle around locally. They are not completely immobilized. This is the actin network, actin cortex. But instead of actually drifting to the right as they did before, they actually sort of move the trajectory to being actually vertical lines along in this chymograph. And so we could quantify that much more rigorously by some tracking algorithms, which then allowed us to extract speed, which we could average over many cells using a sort of a spatial reference here, which is the position from the posterior boundary of the cell. And what you can see is that this particular region, which I mark here, which is corresponding to the region which I showed you in the movie, has actually a lower velocity around actually green, which is sort of 0 microns per minute. So it means that the velocity of this piece of the cortex is being actually immobilized in the wild type. Now, Interestingly, if we look at the integrin mutant in an embryo in which actually cells cannot actually adhere to the vitelline membrane, then we see that the velocity is not actually going to zero. In fact, we even see the opposite, that actually the velocity is first going very positive, because this is due to the fact that the cells actually drift and sort of slide against the vitelline membrane. And then at some point there is a backward flow, which is giving rise to this sort of negative velocity in the referential of the embryo. And so this is what we can see here. And so then we wanted to basically understand what are the two components of velocity, what are the two mechanisms that underlie activation of myosin 2. And what we have in mind is basically first reaction terms, which are dependent on, say, ROC pathway, as well as integrin, as we now imagine. And on the other hand, we have an advection term which reflects the velocity of an active myosin network, in which the myosin motor is actually being advected by the dynamics of the whole network. And so we nailed down the respective contributions of advection and activation by some reaction terms to the activation of myosin 2 in the context of this subcellular wave. And so I plot here in color the contribution of activation specifically in green, advection in blue, and the global concentration rate, which sort of adds these two contributions in red. And so on the plot on the left, what you could see is, a scale bar where you have the distance from the invagination. And on the right, you have sort of a corresponding view at this, about the same scale of a cell, in which you could see the apical surface on the top, and the side view where you could see the cell partly connected and engaged with the vitelline membrane at the top, and then sort of having a curvature because of its detachment from the vitelline membrane. And sort of the dashed line represents the point of detachment, which is to the left. And so what you could see on the quantification is that before detachment, that is to the right of the dashed line, as the cortex is immobilized against the vitelline membrane, there is an activation of myosin 2 by the activation term. There's no advection term, because basically there's no deformation of the cortex due to the adhesion to the vitelline membrane. But later on, as detachment begins, then there is an advection term that sort of becomes more and more important, as the even later the activation, the term is actually inhibited, because the cells are no longer in contact with the vitelline membrane. So this special mapping of activation first, and then later on advection, tells us that first, the activation is taking place against the vitelline membrane, and the advection requires actually detachment from the vitelline membrane. And in this place, activation is no longer taking place. So there's a sequential requirement of the two, which is associated with a spatial mapping on this embryo. And importantly, now, if you look at a scab RNAi embryo, then the activation contribution is basically reduced to minimal levels, reflecting the fact that, yes, integrin is the chief contributor of activation of myosin 2 by a mechanochemical process. And of course, advection is reduced by the very fact that the advection takes into account velocity, the product of the convolution of velocity and concentration of myosin 2. But the important thing is that integrin is required chiefly for the activation against the vitelline membrane. So now let's look at the components of the integrin dimer or complex. What we have is a endogenous labeling of beta integrin with YFP. And at the bottom, you also have labeling of tailin, also labeled with YFP. And so these are specifically recruited to the plasma membrane where the alpha chain is expressed in the propagation zone only. And you can see in these movies, sort of little foci. I hope they come through the Zoom interface. There are small speckles, so you'll see sort of a labeling of the cell outline. But more importantly, you can see small speckles, which are quite tiny. They are not like these big focal adhesions that some of you are familiar with, but rather like small foci. But we could quantify the number, their intensity, how they progress as a function of time. And on the right, you have a typical sort of quantification of the average number of beta chain particle, as we call them, or foci, per cell as a function of time. And you can see that they dramatically increase as a function of time. And on gray, what we show is correspondingly the increased surface area as the cell sort of spread out on the substrate and enlarge the surface to the maximum. Until then, they of course detach, and this when the surface area goes down. So clearly, the integrin foci build up over time during the propagation of the myosin 2 wave through the cell. So I showed you before that integrin is required for activation of myosin 2 at highest levels. And what we have in mind is basically that, let me go back, that the actomyosin contractility network is basically coupled to the integrin via tailin and vinculin, and that coupling actually might be sufficient. That would be the hypothesis one, that the simple mechanical coupling to the adhesion foci to the via the integrins to the ECM in the vitelline membrane would be enough to actually stabilize myosin 2 by a strain-dependent stabilization in the network. Alternatively, there could be some chemical activation from the integrin complexes via FAC or SARC molecules, although at the moment we have failed to identify any requirements for the canonical chemical transducers of, say, activation of Rho. For instance, in a FAC mutant, there's no phenotype whatsoever. And so we are uncertain about the fact that the sort of chemical activation downstream of integrin underlies Rho and myosin 2 activation. Rather, we believe that it might be simply sort of a mechanical stabilization of a network that actually wants to contract, but the flow of the network is actually resisted by the frictional coupling to the integrin complexes, and that is enough to stabilize myosin 2 and increase its activation. And so that underlies the reduced activation of myosin 2 whenever we inhibit integrins. But conversely, and that's the point of this experiment, when we inhibit myosin 2 with a ROCK inhibitor, we looked at what happens to the beta integrin foci, and they clearly dissolve progressively, showing that there was a requirement for contractility to stabilize the integrin foci. So this is shown in the movie here on the right, where you could see that the control on the left and ROCK inhibitor on the right. As soon as we inject the ROCK inhibitor, myosin goes away. And this is a chymograph here. It goes away within seconds. But interestingly, the sort of the integrin particle, which normally form these little tracks on the right here in the chymograph, soon after we inject the ROCK inhibitor, they dissolve and we no longer see any integrin foci in the cell. So clearly, the contractility of the cell is required to stabilize is responsible for the buildup in the intensity of integrin, which I showed you in this particular slide. So that leads us to the notion that there is a feedback between actomyosin contractility and integrins, which is responsible for the activation of myosin 2 from low levels. Sort of these little speckles of myosin 2 that you see in this cell here reflect, we think, and I didn't have the time to go through this, the diffusion of fog, which sort of provides an initial pre-activation of the system at very low levels. On its own, this fog-dependent GPCR activation of myosin to low levels is just priming the system, but is not capable of propagating any wave whatsoever. In order to have a wave, you need to lift this to transit from this low stochastic activation mediated by GPCRs to a integrin-dependent positive feedback that amplifies the activation and allows it actually to propagate through the whole tissue. So that is what happened at the cellular scale. And so in a way, if you map it onto the cell as it actually deforms, you can see here sort of the side view. This is a part of the cortex that is immobilized against the vitelline membrane, where this activation in green is taking place, the positive feedback. This is where myosin 2 goes from low levels to higher levels. But then subsequently, there is a sort of third tier, which is that the cell here in this part is going to be detached or is going to de-adhere from the vitelline membrane because it's being pulled by its neighbors, okay, which pull perpendicular. And so while the shear stress in the plane of the cortex against the vitelline membrane activate myosin to stabilize it and stabilize integrin in this positive feedback, the tensile stress that comes from the tissue scale is going to detach the cortex. And once it detaches, the actomyosin network is going to actually flow, advex more myosin to lift to even higher level. And that is sort of the last stage where the cells actually feed the invagination and basically self-sustain the whole process. And so to address and test the idea that this detachment is key to the whole wave propagation, we decided to test a prediction of the model, which is that if we delayed or affected the detachment we would let the cells being engaged with the vitelline membrane for a longer time, which would give rise to a amplification through the positive feedback of myosin 2 activation and adhesion. And so we did it in several ways, and I'm going to show you one of them, which is to use an alpha-ketinin mutant in which the cell-cell connections is weakened. So think of all the cells in the embryo. They do have a adhesion, but it's weak. And so as a result of that, the cells in the furrow in particular are going to have high apical myosin 2 contractility, but they are not going to sort of team up together to produce a collective contractility that is necessary to pull down and detach the cell from the invagination. And so if you look at the movie on the left, you'll probably see three things, in fact. The first, that grossly speaking, the wave propagates more slowly than it does on the right. This is not here a null condition for alpha-gat, it's a weakening of the expression so that we sort of don't disrupt the epithelium, but actually affect the collective contractility in the furrow. And you can see that the wave propagates more slowly. Moreover, we see second points that myosin 2 levels are higher in these embryos compared to the wild type. So we see the odd case of having slow progression with higher activation of myosin 2. And third, that we see that the cells, in fact, stick more to the vitelline membrane. You see that they sort of form a kind of a ragged boundary because they do not detach in synchrony as they normally do in the wild type. And so we reason that there, what happens is that due to alpha-gat RNAi, the detachment kinetics is delayed. And that is shown in this chymograph by the green curve. The green line is in the chymograph, the speed of detachment, which is at a certain pace in the wild type. And synchronous with that speed of detachment, there is a speed of activation of myosin 2 wave in the cell cortex, shown by the orange line. And these two lines are parallel in the wild type. So if the detachment kinetics is basically timing the wave of activation of myosin 2 by sort of allowing the transmission of the whole activation to a more anterior region, then the delayed detachment will give rise to a delayed activation of myosin 2. The kinetics of myosin 2 activation. And so here are quantification where the myosin 2 wave speed is shown with respect to the cell detachment speed in the different genotypes. And there is a very nice correlation. And it's also the contact time after myosin 2 activation is longer indeed in an alpha-gat RNAi compared to a wild type. And so in this context, then we could look now at the contribution of activation, advection, to the kinetic of myosin 2. And before we go to the curve, let's look at the movie. You can clearly see that in the bottom, in the alpha-gat RNAi, yes, myosin 2 is activated at the most higher levels than in the wild type. And even if you look at integrin on the right, they even go to even higher levels than in the control at the top. So the reason is because detachment is delayed, cells are brought into contact with the vitelline membrane for a longer time. So via the positive feedback loop, both myosin 2 and integrin get amplified and more. And so this you can quantify on the left, where you can see that at the time of detachment, cells remain in contact with the vitelline membrane for a longer time, which is a zone shown in the yellow box. And the activation of myosin 2, if you look at the integral of that below this curve, is much more than in the wild type in the same scale. And that gives rise to a higher amplification of myosin 2. And now, if this is indeed integrin dependent, well, if we knocked out scab RNAi in the context of an alpha-gat, you should have rescued this phenotype, namely that the hyperactivation of myosin 2 should give rise to an inhibition, to a downregulation of myosin 2. And so here's actually, there's a mislabeling. At the top, you have an alpha-gat RNAi, then you have a scab RNAi, and then you have the double RNAi. So the bottom line is that scab RNAi or alpha-gat scab RNAi is the same, meaning that the higher activation of myosin 2 in an alpha-gat RNAi is actually suppressed when you inhibit integrin. So in other words, integrin is basically underlying the sort of delayed or prolonged positive feedback working in these cells. So that leads us to sort of my conclusions, and I will stop there, which is that if we put everything together, first, the conclusions for the second part of my talk is that an integrin-based mechanochemical feedback activates myosin 2. That might be very familiar with things that you might be have seen in cells as they move on a substrate. But there's another scale, which is the tissue scale invagination, which induces the substrate, detachment for the substrate, and propagation of the cell contractile wave. Because if you cannot detach, there's no way you're going to bring more piece of the cortex, more interior, in contact with the vitelline membrane. And so there's no way you're going to propagate the whole cycle of the whole activation of myosin to a zone of adhesion and so on and so forth. And in a way, we could delineate what would be cell intrinsic as well as collective processes that underlie the wave dynamics. There is evidently a local, and I mean by that cell scale positive feedback, which involves integrin myosin 2 coupling. But there's also a collective processes which involve compression of the cytoplasm, adhesion to the vitelline membrane, detachment, which need to be happening in a sequence, in a cycle, to actually allow the wave to propagate. And whenever that is disrupted, then we're going to have a delay in the propagation. And so the whole process of wave propagation requires local as well as non-local collective processes in the embryo. So putting together now where I started, which is sort of the 3 modules of information, namely the genetics, which primes the system and is inherited, as well as geometry, which forms a boundary condition, which is going to be essential for the activation of wave of myosin 2. We could see how then as I showed you, the wave of myosin 2 propagation and tissue deformation is taking place using sort of the initial conditions of the primordium activation of myosin 2, as well as the inherited geometry, which forms a boundary that actually prevents, which actually is going to resist a flow of the tissue and is going to induce by integrin activation of myosin 2. And the second location where actually geometry matters is actually the very invagination of the tissue is going to, through the curvature, to give rise to a detachment, which allows the system to progress. If such a furrow did not exist, if there was no such curvature, there would be no detachment and the whole wave would be stalled. So the wave required the interplay of mechanochemical processes and tissue curvature, which allows for the whole translocation to happen. And I've showed you that when we delay this sort of detachment, then the whole process is stalled. And so I leave with the notion of inheritance, which is essentially, of course, genetics, as we know in chemistry, which is sort of laid by the mother in the egg in the context of development, but also structures and geometry, which actually templates the whole process, and in the way of this boundary drives further development. And on top of this inherited information, there's also novel information being produced through the recursive interactions amongst biochemistry, mechanics, and geometry. And I've illustrated in particular the fact that the tissue curvature in the invagination feeds back on this whole process of mechanochemical activation to drive integrin-dependent wave of myosin 2 activation. So I would like to leave you there. And I would like to thank again Claudio and Anais for having driven this whole project. And I will be taking any question. And thank you very much for your patience.
Thank you so much, Thomas, for such a wonderful talk. We have a couple of questions going on. So I'll take the first one, which you sort of partially answered, but there's a question from Haroon asking what happens, and this is from the first part of your talk, what happens to the wave propagation if we randomly inactivate myosin activation in some of the cells? How would the wave be affected then?
If we randomly inactivate myosin 2 within the wave, within the wave.
Yeah, in some of the cells, because I think you showed like for the overall,
Yeah, exactly. No, that's a very good question. Yeah, because it's true that, so we cannot do this experiment, unfortunately. We can use optogenetics to activate myosin 2 locally, and that is interesting, but we cannot inactivate. We've been thinking, so what we have been developing over the past few years, and I didn't talk about it, is means to actually activate, to use sort of the primordium to activate myosin 2 or FOG, while making the rest of the tissue mutant. So we can create some kind of genetic mosaic, so to say, in the embryo, which allows us to basically address the role of diffusion of FOG, which is important to prime the system, of G-alpha, which is required for cells to actually respond to FOG activation as well as endocytosis. But we cannot inactivate locally myosin 2 or Rho at this moment, and maybe we'll be able in the future, but we can't. Sorry, so I can't answer your question. It's
Wonderful.
A great question.
Next question is from Christian asking, could you comment on the time scales? Because the single cell invagination seems to occur in less than 5 seconds, whereas the time scale of your complete wave cycle is different in your model.
No, the wave propagation, so the speed of the wave through the cell is the same as the speed through the tissue, in fact. So it's a good example where actually, although there are cells, it's almost as if, you could sort of coarse grain it as a continuous medium made of an actin cortex. And even the notion of compression of the cytoplasm, is basically sort of following from the paper of He and Wishouse, where they studied sort of Stokes flow in the embryo, and indeed there, cells almost don't matter. So in other words, the kinetics in the cell and the kinetics in the tissue are the same, and maybe there was a mislabeling, but it's not 5 seconds. It's about a cell per couple of minutes.
Wonderful. Next question is from Yuan asking, nice talk. How does the integrin coordinate with the actin foci formation in this case?
How the integrin coordinate with the actin foci? Well,
I guess when you're looking at that.
Yeah. So there's one thing which is striking in the wild type and also in the alpha-catenin is that if you look at an ester of actin and myosin, we see that the foci of integrin are actually sort of forming like a crown around the actin foci. So as if so we know that in other cases in the fly embryo, you have spontaneous generation emergence of an actomyosin pulse, which has sort of centripetal flow. That's what Akankshi characterized when she was with us. And so in the case of these specific cells, that is taking place, but it's also sticking to the vitelline membrane. And so that means that that explains why we actually see the foci of integrin as sort of as a ring around it, because it resists sort of the centripetal flow. Mm-hmm. And so the geometry of the foci reflects the topology of the flow, which is centripetal. So that we see very, very conspicuously in the case of alpha-catenin, because everything is boosted up. And the wild type is a sort of like that, but the levels are a bit lower to characterize.
Great. Next question is from Esteban says very interesting. In the side view, there is an in the side view of the images you showed, he could see an accumulation of actin in the region of compression. And he's asking, is there a force contributing to the process in that region?
So accumulation of actin
in the region of compression, yeah.
I'm not sure this is necessarily true. I don't think we see an increased density of actin against in the region where integrin base adhesion is taking place.
He did put, I think, so it could be his observation.
Yeah, maybe there was. So if you look on the side in the 2 photon, there are some kind of optical aberrations due to the fact that it's a Z stack and so on. So I would be more convinced if you look from the top with spinning-disc confocal, and typically Absolutely. I don't think we've seen any such thing. So I rather believe that no, there's no change in density, but there could be some local, inhomogeneities or increased density. I mean, you could think of, but the other thing I should say that we have not characterized in this context, regulation of actin nucleation, although this is something that we are going to investigate in more detail. There could be some local actin regulation due to the fact that integrin is, of course, being activated. In particular, I should say that if you look, I mean, it was hard for you to see, but if you look at the cells as they flattened, at the rim of the cell, there are some kind of lamellipodial protrusions in which I think you could see sort of kind of retrograde flow. Mm-hmm. And I think that where the cells, they're stuck by the apical surface, but the rim might be analog to what has been reported in many cells that actually plated on a substrate that they actually have local integrin-dependent spreading. Yeah.
That's very interesting. So there's a question from Ruth Johnson asking, do you think the pole cells contribute to this process at all?
No, they don't. So you can, so the pole cells that are in the primordium, they are sort of sitting above the primordium. When you remove pole cells, the whole flow, symmetry breaking is still taking place. And we've not specifically characterized the wave in these mutants, like Oscar mutant, for instance, but we know that the flow, the symmetry breaking is taking place, and I and I don't think anyone has reported, we have not seen any gastrulation defects. So I don't think they're required. Yeah.
Perfect. If you're okay with taking a few more questions.
Yeah.
The next question from Anna Pasapera asking, it has been demonstrated by several groups that vinculin is recruited in a force-dependent manner in integrin-mediated attachment as well as ecadherin cell attachment. So did you look if there is a differential distribution of vinculin as a mechanical sensor in embryos lacking integrin and catenin?
Yes, so precisely, that's true. So we've looked at vinculin. And in fact, we had looked at vinculin before we knew there was anything like a wave or whatever. Integrin, and actually, Girish had actually remembered that there was something going on. And he thought that these apical surface speckles of vinculin was an artifact. And then we looked back and we said, oh, interesting enough, it's there. So if you compare talin and vinculin, the remarkable thing is that vinculin is specifically recruited in those regions near the detachment zone where myosin 2 levels are highest. So in a way, talin is recruited at sort of broader levels in the speckles in regions where contractility is boosting, is increasing, but not very high. And vinculin is recruited later on. I think this is consistent with what people have reported, that it requires higher tension for it to be recruited. But as you probably know, the vinculin is not actually, sadly enough, so important in flies. In flies. And so, there are other mechanisms to sort of make mechanical, stabilization of the bonds than vinculin. But it is definitely behaving as a perfectly normal and important molecules. It's, both at junctions and in this sort of in the plane of the cortex.
Yeah. Wonderful. Next question is from Ja asking a knife question: how the buckling ectoderm decide when and where to stop invagination or the wave?
I think the stop signal is probably reflecting the fact that so you have an invagination going on. And of course, the anterior movement, the flow, and the wave are resisted by the mechanical compressibility of the whole tissue, the anterior region of the embryo. So at some point, you may be increasing the pressure that is going to resist any more anterior movement. The other thing also is that at some point you run out of fuel, because what fuels the invagination and what sustains the whole process is the fact that you have a flow or flux of cells from the dorsal epithelium that actually goes into the furrow. So long as you add more cells, you sustain the anterior compression and the whole cycle. But if at some point you run out of cells, you're going to arrest everything. And indeed, at some point, you've advanced the whole thing so much to the anterior that you kind of run out of cells. And so the system would go to a halt. So that's why, basically you have a sweeping wave that goes all the way to almost a cephalic furrow, and then it stops. That could be the other reason. It could be a combination of the two.
Wonderful. Next question is from Debashis, which I guess other people are also wondering, asking question on the importance of geometry here. Is the elliptical shape important for the production of tissue curvature and wave propagation?
Yes.
Or any confinement would do the job?
Yes. So this is actually another talk. But I decided not to talk about that, but that's super important. So yes, we have a story that we'll put on bioRxiv hopefully soon, where I actually address specifically the flow. I sort of put under the carpet this extent of the flow that is required for the buckling, the invagination, and then everything that I told you about. So what gave rise to the flow and the symmetry breaking? So it turns out that depends on geometry as much as contractility. And in fact, it is the mismatch between a pattern of contractility and the gradient of curvature of the embryo, which is not, which exists because it's a kind of elliptic or ellipsoidal embryo, which actually is breaking the symmetry and allows the flow to progress. That is true in 2D. It's true in 2D, and we think might be even true in 3D. But the essential conclusion of that story is that to break symmetry, you need to inherit geometry and contractility. So which is exactly what I already said, but this is illustrated in the context not of the wave, but of the flow. So geometry is super important. Yeah. So in particular, if you make an embryo that is more spherical, which we've done, less round, then you affect the flow in a way that is predicted by the model. So you'll see these papers hopefully soon. But yeah.
Awesome. Next question is from Tianchi Chen asking, what happens to the integrins after cell detachment? Are they still bound to the ligands or remain activated, able to promote myosin contraction?
So we don't know. So what we know that the ECM is actually, the vitelline membrane is made of this collagen 4 in it. We don't know the full composition of the ECM. We're actually thinking of actually doing a very simple experiment of actually using collagenase to see whether we could actually soften the vitelline membrane and maybe change the boundary conditions and allow for other geometry to emerge. It's so simple that we didn't think about it. It was actually a student at NCBS that suggested we might do this, and we will do it. So we don't know whether many ligand might be actually taken as the cells actually are detaching. For various reasons, we become increasingly interested in ECM. In a way, the ECM in the embryo would be probably the case of a very stiff, ECM that cannot change its structure. The cell would basically get in contact with them, and that's it. But we know basically nothing about the vitelline membrane. It's an unexplored field, and we need to learn more about that.
Wonderful. Next question is from Jen, whose mic is not working today. So since you see lamellipodia, is there any sense in which these cells are crawling on the vitelline membrane?
Yeah. So, my title would have suggested that they might do so. You realize that I'm talking about adhesion, where actually the engine of the motility is not actin polymerization at the leading edge. Really. It might be important for the spreading together with the fact that you have a sort of a tissue level mechanism to actually spread the cells against a sticky surface, right? So that's another case where you have probably local phenomena as well as non-local tissue scale phenomena. But apart from the fact that the engine is not really, actin polymerization at the leading edge or actin retrograde flow. To be honest, early on, I thought, and I betted against Claudio and Anna, is that it might be just simple collective actin retrograde flow that is going to be resisted by some friction or adhesion that drives the whole process, but it turns out not to be the case. But the point is that nonetheless, if you think about the analogy between what I reported to you and cell motility, in cell motility, you have an actin polymerization resisted by friction, adhesion to some substratum, and then some detachment at the rear, which is myosin 2 dependent. And that is coupled to the front in a way. You still find, and you have adhesion, de-adhesion kinetics driven by some kind of by some kind of contractility. The big difference between simple cell motility and our system is that the active process is not really cell actin polymerization, but rather some kind of compressive force driven by a posterior process.
Yeah.
But nonetheless, the idea of a cycle of adhesion, de-adhesion driven by contractility is taking place. And so it's interesting to think about the analogies and differences between what we call cell motility and its various forms and here. The other thing is that I've not seen much studies of cell motility on substratum where actually geometry, curvatures might be, an essential ingredient. Usually people use simple substrate to do turf and do all of that. That's great. But, in vivo cells don't care. They go any bumpy regions and they will use amoeboid as well as a substrate kind of motility. How do they cope with curvature and all the environment that I explicitly, talked about in a case where it's actually very well controlled? I think it will be an interesting area to investigate, even at the cell scale. And so actually now, we're going to actually not only use experiment, but also use as a model with Mud and Rao at NCBS, actually use a model that actually encapsulates the geometry of the confined space where fog diffuses, GPCR activation, mechanical processes of the cortex, and the geometry, which itself adjusts the space available for diffusion of fog. So it's an interesting case where we could use modeling to actually address the interplay of chemical processes and mechanical processes together with geometry. Because you see, the geometry that I'm referring to, curvature and space for diffusion, are themselves the product of the very active contractility that I talked about. So they're all dependent upon one another, as I said in the beginning.
Wonderful. There's a question from Timothy Cox asking, related to the question about that, is there a constrictive force related to the cuticle that has to be accommodated in the invagination wave model? I think by cuticle, he probably meant the...
So can you repeat the question? Sorry, because I was confused.
So there is a there's a constrictive force. Is there a constrictive force related to the cuticle that has to be accommodated in the invagination wave model? I think he means.
Okay, so I think how I understand the question is, so if the cuticle would be the vitelline membrane.
Timothy, if you're around, you can also ask the question.
Yeah, I'm not sure I understand the question actually. Sorry.
Let's see if he's around, he will pop in. Okay, the next question is from Amit Das saying, great talk. Thank you, Thoma. Have you observed any loss of cell-cell coordination at the trailing edge in alpha-cat RNAi? I mean, the coordination in terms of correlations between the cell velocity vectors.
So we haven't done any quantitative correlation analysis, but what you've seen in the movie, and I did highlight that, it was sort of ragged boundary. The cells did not detach in sync. So think of a tape, a sticky tape, in which just because you've removed the force, when you have a tape and you want to detach it, you're actually building normal stress to detach. That is actually what is happening. So now, if you reduce the normal stress, like in alpha-cat, the material, that the tape that you detach is made of many cells. And so some will stick more than others because, the cells will actually accumulate more and more myosin too. So the reason why some cells stick more, we think, is because the positive feedback keeps going, and some cells may actually activate myosin to more than others. So the expectation, which we haven't tested, is that the irregularity or lack of correlation would actually be predicted by the extent of how much myosin 2 is being induced there and how much integrin is being further activated and how much more sticky the cells are. There's a nonlinear amplification, and so that's how you could easily shoot too high in adhesion, and then it's going to be harder for you to be detached. So that's something which we have not tested quantitatively, but could be.
Wonderful. I think that's the end of the questions. If I didn't miss anything, Jen. That was a lot. I have one of my own.
So that was a good sequence of questions.
Yeah, it was wonderful. I have a question of mine. I was wondering, because this integrin regulation is clearly under transcriptional regulation. Do you know something? Why is it specifically just at that point? And like rest of the embryo doesn't have that sort of attachment.
Well, if you express integrin everywhere in the embryo, we've done it. Well, what happens is that you make spurious adhesion to the vitelline membrane, and a lot of things, and the whole process is gone. I mean, gastrulation is blocked.
Interesting. So you need that space. Yeah.
Well, think of cells. They are going to have a surrounding envelope. You don't want them to stick too much. You actually want them to flow in general, right? You don't want to have any friction or adhesion that would actually affect the flow, which happens everywhere, like German extension. It's all of that. So what you want is have very selective adhesion, and so that's probably why scab is in a specific domains. And if you don't have scab at all, if you have no adhesion, then you have other problems that you cannot make this expanding invagination. So one thing you could ask is, what do you have not such a bigger domain that invaginates all at once? It just doesn't work. It's probably the case that to make a deep invagination, you have to initiate it somewhere and then propagate it in a wave. And that's why you need a pattern domain of scab. And actually, these are the reasons might be actually geometrical. If we use simulations where we have a big domain for invagination, it actually quickly gets stuck into some kind of a conformation where the pressure in the yolk is actually affecting the further development. Well, if you do it in a wave, you actually do it better. These are things that we are going to test.
Yeah. Oh, you have a question from Akanshi from YouTube.
Akanshi.
She's asking if the tangential question on the evolution of furrows, you partly answered that already. Can you in your model predict the position and depth of buckling in eggs from different insects based on size and curvature?
Yes, if we had a good model that actually encapsulates geometry, mechanics, and all of that, then we will be able to explore sort of the variation across species, but we are not there yet. But absolutely, it would be an interesting thing to look at. I think that broadly, I would like to finish maybe with that statement, is that the vitelline membrane is the last thing we thought about in terms of being important. You're probably familiar with the work of Pavel Tomanchak and Stefan Girls, where they actually show that in tribolium, the vitelline membrane is used as an anchor. It's a fixed point. In our case, it's a dynamic adhesion zone. In chicken embryos, the epiblast spreads, and its substratum is actually vitelline membrane, which is probably very different in terms of composition. We don't know, but their integrin actually is required at the periphery of the epiblast to expand. So I think it's been a largely overlooked area, and it will be interesting to consider this as the ECM, the vitelline membrane, is a substrate for interaction during early development.
Wonderful. I think we are done.